Assalamualaikum warahmatullahi wabarakatuh and good evening. Today, our group would like to present our title on data mining and analysis of color-related gene in fishes. So, this is my group member. For introduction, the Atlantic salmon, which is Salmonella, and the 7 species of Pacific salmon are members of one of the most primitive superorders of the Teleos, namely the Protocanthropteridae. The family Salmonidae comprise the Atlantic and Pacific salmon, the trout, the chars, and the haken. Typically, native salmon populations are genetically distinct from each other and adapted locally. Farm and wild salmon vary in a variety of characteristics, including genetic polymorphisms, development, morphology, history of life, behavior, physiology, and genetics. Color is important characteristic of quality in salmonids. While salmon pigmentation is derived from the absorption and deposition of dietary oxygenated carotenoid. Astaxanthin is the primary pigment, occurs in white salmon at a concentration of 3 to 11 mg per kilogram and 4 to 10 mg per kilogram in farm salmon. Color patterns are a striking characteristic of species, changing rapidly and playing major roles in both natural and sexual selection. They act as camouflage in predator avoidance and often remarkably elaborate and have an important role in kin identification and mate selection. And this is example of the Salmonella. Objective of this assignment is to analyze gene color by using Swiss model to create 3D modeling, and then to obtain the correct sequence from the NCBI. Next, I will pass to my group member to explain about material and method.
Okay, for the material and method, we use FASTA formal sequence, accession number EXP01398251.1, which is a predicted melanocyte stimulating hormone receptor for the Salmonella species. The procedure is first we use the program practical Swiss model. Then we copy the sequence Salmonella and pass at the Swiss model. Then proceed by click the build model. Then we wait for the few minutes or seconds. Then we can get the result. Then record the result for the reference. Okay, this is a software which we have been used, which is Swiss model. Okay, first we copy and paste the FASTA sequence of the melanocyte stimulating hormone receptor, which is Salmonella species. Then we go to start modeling. Then we paste the sequence and we click the build model, then wait for the few minutes. This is our result, and we can save the information and screenshot the picture for the 3D model, and we use it for the result information. And this is the material method for the 3D model structure.
We choose Swiss model because we want to see 3D structure of protein sequence. Swiss model dedicate protein structure homology modeling. It build protein homology model at different levels. Swiss model modeling 3D structure of protein based on its amino acid sequence using homology modeling technique. Beta 1 adrenergic receptor and cytochrome adenosine receptor have protein homology model with amino acid sequence from given sequence. The two models show one of the receptors involved in the nervous system. Identification percentage for both sequences are 29.52% and 27.44% respectively. Swiss model also show the 2D structure by separate position of hydrophobic bond and protein subposition after those Swiss model.
For discussions, the protein sequence that has been used in this laboratory is melanocyte stimulating hormone receptor or Selmo-Salar. From sequence, we can get the information for Q mean, which is negative 4.82, GMGE which is 0.48, and for the sequence identified is 29.52%. Based on beta protein sequence, the result shown by Swiss model is the sequence for 3D modeling protein structure of soluble cytochrome adenosine receptor for species Selmo-Salar, which is beta-1 and adrenergic receptor and soluble cytochrome adenosine receptor. The beta protein sequence, we can get the information for Q mean is negative 4.82, GMGE is 0.48, and sequence identified is 29.52%. Their description of the predicted sequence of Selmo-Salar and two are same, which is beta-1 adrenergic receptor is Turkey with stabilizing mutations and bound endogenous carazole. They are slightly different in the 3D model between 3D modeling protein structure of beta-1 adrenergic receptor and 3D modeling protein structure of soluble cytochrome adenosine receptor. Even they are from a same sequence protein, the 3D model show they are different but have a same Q mean value, GMGE value, and sequence identified percentage.
So now I want to state the problem we had when we do the assignment. So the first problem is we are not familiar in using the NCBI and we are hard to confirm the correct sequences for the coloring genes in a fish species. Second is not able to meet face-to-face for assignment discussion during the MCO in Malaysia since the virus COVID-19. The situation makes us hard to work and discuss about the assignment. Next is there are many confusion between the coloration gene and coloration term. We are hard to determine the differences of coloration genes and coloration term. So the solution for the problem we use to do is we use a video prepared by lecturer and YouTube video from internet as the reference and guide in using of NCBI. So and we also make a search on the article and book that explain about the coloration genes and coloration term. So another solution is the faculty also can make a poster about the using of biotechnology in fisheries, especially in NCBI application, to give more information to student in the faculty. Last but not least is make a report or slide about biotechnology advancement in the aquaculture and fisheries industry in Malaysia and the world. That's all from me. Thank you.